Good afternoon, and thank you for joining us for this Outdoor Art Club presentation, Finding Art in the Ocean. I'm Kate McCormick, and I'm collaborating with Diane Wagner of the Outdoor Art Club's Zoom team to bring you today's program. The Outdoor Art Club is a nonprofit women's organization founded in 1902, and our mission is to make civic, cultural, and charitable contributions to our community. Every year, we present a public speaker series so that people like you have the opportunity to learn about topics as varied as affordable housing, to understanding modern art, to learning about the wonders of seaweed. Our speaker, Josie Iselin, looks forward to answering your questions at the end of the program. Please feel free to enter questions by using the Q&A function anytime during her presentation. On many computers, the Q&A button is found on the bottom of your Zoom screen. At this time, I'd like to introduce Kathy King, who is a member of the Outdoor Art Club and program chair for today's event. Kathy, thank you for bringing us today's program about the fascinating world of seaweed.
We are so pleased to welcome artist and author Josie Iselin, whose newest book is The Curious World of Seaweed. In this beautiful volume, which was published by Heyday Books and released August 2019, Josie explores the art and biology of 16 seaweeds and kelps that live in the thin region where the Pacific Ocean converges with the North American continent. She believes that seaweed can teach us about their resilience, their poetry, and magic. Josie will describe her path as an artist into the science of seaweed, highlighting how several of these foundational organisms succeed in their ocean niche. I think you will agree with me that Josie's particular, spectacular, and colorful imagery, which she has collected from explorations along our California coast, illustrate the science and wonder of this ocean universe. Josie holds a BA in Visual and Environmental Studies from Harvard and an MFA from San Francisco State University. She currently teaches in the School of Design at San Francisco State. As a fine artist, Josie exhibits large-scale prints at select galleries, museums, hospitals, and other spaces.
Thank you so much. I thought what I'd do is start us off with an introductory video, and then we'll go into my talk from there.
Seaweeds and kelp are some of the great eco-engineers of our planet. They are photosynthesizing powerhouses, growing rapidly in cold waters, creating the base of the vast ocean food chain. Kelp and seaweeds provide crucial habitat for countless other organisms, both tiny and large. They sink carbon and oxygenate their nearshore waters. Marine algae, another name for seaweed, are essential and also spectacularly beautiful. Their stories are compelling and important and deserve to be told with more than words. My newest book, The Curious World of Seaweed, begins like this. The thin region where the sea meets the land is unlike either land or sea. It is betwixt and between, a threshold from one state to another. It is a linear point or a coastal ribbon, a place of dramatic change and remarkable abundance, abundance of life and also of possibility. From the ocean's perspective, the approach to the world's great land masses is from the deep and dark pelagic, up over a continental shelf into a dim region where light barely penetrates the water, and on up into a brighter photic zone, what might be called the subtidal, and continues into the low intertidal where the extreme full moon tides pull away the water once or twice a month, and up towards the beach or rocky reef, where the ocean swells in and out in 6-hour cycles, and up finally to the highest tide mark, the rack line, where the ocean leaves its debris to mark a final encroachment towards dry land. This sliver of ocean, where sunlight penetrates enough to allow photosynthesis to work its magic, and where the benthos or ocean bottom provides something to hold on to, is the home of the seaweeds or marine algae. The Pacific Ocean's edge, where it encounters the North American continent, is considered one of the richest of these rich zones. This slice of ocean, from Alaska to Baja California in Mexico, has some of the most diverse and abundant seaweeds and kelps on Earth. The rocky, fog-shrouded coast sports a spectacular number of seaweed species, from enormous kelps to tiny corallines. The portraits and artwork made using my flatbed scanner have taken me deep into the world of seaweed, initiating a journey into scientific storytelling, ocean advocacy, and algal empathy. I hope you might come with me.
Thank you so much. And I am just absolutely delighted to be here with the Outdoor Art Club. I'm really looking forward to bringing kind of this world of the seaweeds out to you who are so concerned with our natural world, and to kind of try and take us into other spaces, other places, rather than our kind of land-centered universe that we feel pretty comfortable with. So I'm going to share my screen and take you through my imagery and my kind of journey as an artist into the science of seaweed. We will share the screen there. I hope that is working. So my mission has been for many years now to bring the world of the algae out kind of to a broad audience. And it's my artwork that has really led me into the science of the seaweed, into the research that has taken me into the writing that has been then combined with the artwork to make a series of books. And I'm also always showing work in various different gallery scenarios or public spaces. This is an installation at a gallery in Alameda. And what I've done is printed some of my imagery of these various seaweeds. We have Erythrophyllum, Egregia, Palmaria, and Macrocystis. I've printed them as 94-inch high curtains. These curtains have allowed me to kind of bring these organisms into unsuspecting places, not only galleries, but this is actually an installation at a very underused shopping center, again, in Alameda. And the Alameda Arts Commission commissioned a bunch of us artists to enliven the shop fronts of very underused storefronts at a shopping center. So I'm able to capture these seaweeds that are, as I mentioned in the video, both tiny and large, and create different spaces to encounter them, whether it's the pages of a book, the walls of a gallery, or the windows of a shop, because my tool of choice is my scanner. So this is how I capture this imagery. And I've been using my scanner for probably 25 years, really a long time. And scanner technology does not change that much. So this scanner is right here in my studio. I'm kind of facing you and my computer, and right to the right of me is my scanner. And what you see there is a seaweed specimen that's on the glass. And there are kind of two ways to use a scanner. I can scan opaque things like those stones that are sitting out front, and I would place them carefully on the glass and leave the top up and let the scanner actually bounce light, reflect light off an opaque thing. Or in the case of the seaweeds, I want to capture that translucence, that's transparency. So what I do is I use the transparency adapter, which is the top that's right now is open, and I take off that white cover. And up there is a second light element. And that second light element, once I lower the top down, either all the way for a very flattened object, or if my specimen is somewhat flouncy and three-dimensional, I'll use my helper stones there to prop the top up a little bit so it doesn't squish it. And then that second, the top there, that second light element pushes light through the specimen as the scan is captured. And so this scanner gives me not only very true color, and very consistent color, which is really important in the seaweeds. And you know how hard that can be in the world of seaweeds. But it also gives me an enormous amount of resolution. So that means I can make these images very big. So for these installations where I'm printing on fabric that's 94 inches high, I have the resolution to do that and have these crisp, beautiful, dynamic images. So this is again back at that Alameda installation. You have the bull kelp or Nereocystis on the left here, and you have Palmaria on the right. I'm very insistent on naming these seaweeds so that even if we're not comfortable with the Latin names, we at least start to get them in our ear. And there was a whole festival at in this shopping center. There were children's performances and dancing and food trucks. So there was a huge mix of people that really would never get out into a kelp forest that get to encounter these organisms. And I'm very aware of that we can't, most of us can't hike into the kelp forest, not the way we can hike into our, on Mount Tam or go to the redwoods or what have you. So, so I really love to collaborate with the scientists who are experts in the seaweed. And so seaweed scientists are called phycologists, not psychologists, but phycologists. And part of their passion that got them into the science of seaweed is how beautiful the seaweeds were when they first encountered them. And so they really love getting me involved. And so this was an installation for the conference for the Northeast Algal Society back in 2018. And their mantra for this conference was broaden your impact. So how can they get their world of the detailed research that they do out to a broader audience? And that's where my artwork comes in. And it was really fun to work in this kind of funky gallery space and really bring it to life with these seaweeds. What you have here on the right is a brown sausage-like seaweed called Cytosiphon, and the one next to it is the sea grapes or Botryocladia. And so I did a whole talk in that space for the introductory meeting of that conference. So the scanner is really good at stripping away context and letting these organisms really speak for themselves. So how I initially really focused on using the scanner was to do these very straight ahead portraits of the marine algae. So that's what you have here on the left with this beautiful specimen of erythrophyllum. And erythrophyllum is very common red seaweed out on our California coast and up into British Columbia. It really is that rosy red. It's in the red category, and I'll talk a little bit about those categories later. But you can see that it also has a little bit of the ocean still with it. So I can scan these specimens right after I've collected them, and I kind of pat them dry a little bit, but you can still see that wetness, that a little bit of the ocean that's holding on there. Or I press them in a plant press and scan them dried. And after my book, An Ocean Garden, came out in 2014, I started experimenting with these more elaborate collaged kind of constructions where there's more abstraction involved. But I always like to let the specimens be able to be identified. This is a group of the wonderful Halosachion or sea sacs. And sea sacs are very common out on our rocky reefs. They start sprouting up like these fingers, which you can find at low tide, especially in the springtime. So at the springtime low tides, it's worth going out to the tide pools. And these Halosachion are interesting. They're known as sea sacs, and they've taken a different tactic towards survival in the intertidal zone. And we have to remember that the intertidal zone is that place where these seaweeds' world, their atmosphere, so to speak, is the ocean. And the ocean gets sucked out every 6 hours and comes flooding back in. That's the tide. And we can't quite imagine existing in that kind of a changing world. I mean, imagine if our atmosphere here just got sucked out every day and then came flooding back in. Well, that's what these seaweeds have to contend with. And many of the folio seaweeds are very good at drying out and rehydrating. But these guys actually hydrate from within. They have whole little oceans within them, so they stay hydrated during the low tide. They kind of stand up like fingers. They have pores up at the top where the water can go in and out. And they are also in the red category, even though they present as this incredible celadon green. This is Gloeosiphonia, and it's a scan of two historic pressings made in Monterey in about the 1880s. And then I've overlaid it with some other color from other seaweeds, a little bit of artistic license there. But it's to show you the incredible array of forms and qualities that the seaweeds present to us. This feathery quality is very different than the leafy seaweeds. And these forms for me as a designer are an absolute delight to work with. I get to design the page, I get to design spaces. You have this wonderful Maziella volans, which really does have this spoon-shaped quality and dries to this amazing purple color. And I've gotten to pair it with these wonderful and whimsical bladders or pods of the egregia or feather boa kelp. And that whimsy, that delight in design is really me just looking and exploring what the seaweeds are presenting. So Egregia menzicii, named after Archibald Menzies, the great naturalist who was on board the Vancouver expedition in the 1790s, is commonly known as feather boa kelp. And this is a very, very common kelp on our California coast. It's actually one of the kelps that's doing quite well with our warming oceans. It's not one we kind of have to worry about. But it was when I was writing or kind of developing the proposal for the book, The Curious World of Seaweed, I challenged myself with writing an essay titled Empathy for a Kelp. And I really challenged myself to kind of get us to understand or have empathy for the feather boa kelp, because it's so different than anything that we understand in our terrestrial world. It doesn't have to combat gravity. It has just this weird shape and form that doesn't translate into anything that we recognize. And it also has this incredible variability from specimen to specimen, depending on how old it is and where it's actually growing. But it is only one species, Egregia menzicii. There's only one species in that genus. So it can have this kind of thin strap with these spindly blades that go off of it, or it can have a very thick strap with these paddles, these kind of thicker paddles that come off of it. And I, as an artist, really have taken inspiration not only from the organism itself and all this variety and organic shape that it gives me, but I've also taken inspiration from other artists. And I don't know whether you all remember that amazing show that was at the SF MoMA a few years ago now, the Matisse Diebenkorn show, where it really presented kind of Diebenkorn taking direct inspiration from Matisse. Well, I went and saw that show about 3 times or so and thought, you know, to kind of spark some of my creativity and I needed a jolt for some new art making. I took inspiration from one of my mentor artist, Rex Ray, who's a San Francisco artist who was very much walking a line between graphic design and fine art. And Rex Ray would cut out these fabulous organic shapes from this pattern paper that he would make. And then he would make these spectacular collages from those shapes. And I thought, well, what if I use the blades of the egregia itself as my organic shape? And then I collage with that. So this is called Rex Ray number 6, and it's one of a whole series that was inspired by this way of working, and with these different specimens of the egregia that I'd been collecting. I've also taken a lot of inspiration from Anna Atkins, and Anna Atkins was a polymath from the Victorian era, and she experimented with a very nascent photographic technique called cyanotype printing. And I decided to, in her honor, to do some cyanotype printing myself, which is how these are made, with my specimens exposing in my backyard right here. So cyanotype printing involves coating paper with a light sensitive emulsion. And that emulsion, once it dries, once it's taken out into the dark and exposed with one of your specimens, one of my specimens on it, what happens is that the UV from sunlight, once you've done the exposure and you rinse it out in water, that where the UV has hit the emulsion, it hardens and it washes to this deep Prussian blue. But where you've been shadowing it with your specimen, the emulsion washes off and you get this the shadow of your specimen. And so Anna Atkins is, been pretty well written about now, but in about 2009, when I started researching her, she really, it was hard to find information about her. And she has, the more I get to know about her, the more inspiring she is. And I realized that my scanning techniques of using my scanner is actually in this lineage of what's called nature printing. And nature printing is where the image is made directly from the specimen. It's not mediated by the hand of a botanical illustrator or a scientific illustrator, or it's not mediated by the lens of a camera or the eye of the photographer. It's really the specimen itself that is creating the image. And I thought, oh, I love that my scanner is kind of in this same line, and couldn't my scans actually work in dialogue with the specimen's shadow made as the cyanotype print? So I started laying my scan into its own, the scans into its own shadow. And this is, this is Picea californica, and this is a Puntiella, also californica. And this is a fabulous red seaweed with these kind of mouse ears. This is quite a mature specimen found up at Limantour Beach. And this playfulness between the specimen and its shadow is what I'm kind of after. And also a dialogue between past and present, between my contemporary scan and this historic printing technique. And this idea of dialogue between past and present really has it's like a vector going from past to present, and a vector has an arrow on it, which is heading out into the future. So there's this premise or this question that really undergirds all of my work, whether it's the art making, the research, the writing, is where will these organisms be in our changing, given our changing oceans, with the warming oceans? And that is just an ongoing question that I think we constantly have to ask ourselves, and what can we do to kind of mitigate that warming? So in 2014, I published An Ocean Garden, The Secret Life of Seaweed. This was my first book exclusively on seaweed. I'd been doing a number of workshops with the great Kathy Ann Miller, who's the curator of marine algae over at UC Berkeley's collection that's part of the Jepson Herbarium. It's called the University Herbarium, and it's just the algae. And I learned so much from her. And once I published this book, which immediately went out of print, needless to say, but can still be found through my website or through any of the used book sales outlets, I really wanted to continue looking into the story of the history of the science of certain of these iconic seaweeds that I was finding on the beach. And I wanted to know more, and I wanted to delve into. And I realized that the history of the science, the taxonomic history, the history of its naming, how we know what we know about each of these iconic seaweeds, has this really robust visual component to it. So these are 4 lithographs that are from a pamphlet that was the first pamphlet of 1853 describing these kelps of California. It was written by Franz Josef Ruprecht, a German botanist working in Russia. And so on the left, you have Stephanocystis osmundaceae, you have Dictyoneurum californica, you have Pterygophora, and then you have the great Egregia menziesii, or feather boa. So these are 4 of the 6 species that were described in that pamphlet. And these lithographs are spectacularly detailed. And Ruprecht decided to show the whole organism in describing the visual description of, and so he had to paste together these panels, and they're folded up in the back of this pamphlet. And so when they're unfolded, the Egregia is probably 5 feet tall. It's really spectacular. So again, I really wanted to experiment with my contemporary scans in play with these historical documents. And this was one of the first combinations that I did of this old, or new on old, as I call them. So this is Costaria costata, a scan of the Costaria costata on a lithograph of its cousin, the Dictyoneurum californicum. So there are a bunch of seaweeds that have this texture on their blades, the kelps, and it's actually a rugose, it's called a rugose texture. And this texture creates a micro turbulence at the cellular level. So these foliose blades are all trying to maximize their surface area to volume, so that because each of the cells on the blade not only photosynthesizes, but each of the cells actually collects nutrients from the ocean around it. So that microturbulence at such a micro level is bringing new water with more nutrients to each of those cells along the blade. So that's kind of cool. And rugose is a great word to this cabbage-like texture that this particular seaweed has. This is a scan of nori or pyropia, which is very sheer. It's only 2 cells thick. And I've combined it with a lithograph of nori by the great Alexander Postels. And nori is actually in the red category. So Postels' rosy pink nori or pyropia behind there is very appropriate. And Alexander Postels was one of the naturalists on board a Russian exploratory expedition to Alaska in 1829. And he was brought on as the surgeon, or I think he was trained in geology, but he had this incredible affinity for drawing, number one, and he had a real affinity towards the seaweeds that they were finding up there in Alaska, which has an incredible flora of seaweeds, many of which, many of the flora actually extend down into Northern California. So Postels actually paired up with Ruprecht, the botanist, to make this incredible publication published in 1840 called Illustrationes Elgarum. And it's a big oversized folio where Postels' lithographs are combined with the descriptions by Ruprecht. And there's a copy of it in the libraries at the University of Southern California. So I got a chance many years ago to go down there. I made my daughter look at the school. She had no interest in it, but I was like, you have to go look at this school so I can spend the day in the library. So here are some of my scans of nori or pyropia found here in Northern California on the coastline. It's very common, and it's really what indigenous tribes would collect into patties and store, dry for the winter and store and eat. It's very delicious. And it comes in this real range of color. So you can find it as dusky green here on the left. Some of its reproduction is happening on the edges there, or it can be found as this deep purple, almost black. It can be so purple. Again, the seaweeds just present so many different kind of sides of themselves to us. And this is the ulva or sea lettuce. And ulva is really common again on our rocky reefs and on our beaches and seashore here. The sea lettuce is really the signature seaweed of the green category. And the greens were what gave rise to plants on land. I'll talk about that in a minute. But this is again combined with lithograph by Postels. And here you have the beaded rack or Stephanocystis combined with its Ruprecht drawing there. This became the cover of the book, The Curious World of Seaweed. So I have this great process where I not only make the images, I research and write these chapters, but I'm building these books as I go along, combining the word and image and making sure the sequencing of things is really dynamic. And then I send that rough outline to the art director and the designers at whatever publisher I'm working with. And in this case, it's Heyday Books over in Berkeley, and they're just a fantastic publisher, and I had such an amazing experience working with their designers. So there's 16 chapters based on 16 iconic seaweeds that you will find on the coast. They're not rare ones. There's one that's a little bit Hardwicksia you probably wouldn't find, but the others are really standard iconic seaweeds. And these are only just a tiny portion of all of the seaweeds and the flora of our California coast. And I just want to, kind of reiterate that. And this abundance of the marine flora on our coast is due to the fact that we have cold ocean cold ocean is so important for the health of these nearshore ecologies and for these primary producers, the seaweeds. And that cold ocean is really every year during spring, we have the northwest winds come in. And this is what I'm kind of showing here. This was taken in the springtime on the Mendocino coast at McCarricker State Park. And those northwest winds come and push the surface waters away and allow the deep waters just beyond the continental shelf, which is right near our coast, to come up or upwell. And these deep waters not only are very cold, but they hold a tremendous amount of nutrients. So that cold, nutrient-dense ocean that comes up to fill the space on the surface is what allows this abundant growth, especially in the springtime. And this abundance can really be seen on the shore by us beach walkers. You know, as I mentioned, I'm not a scuba diver. I do snorkel whenever I can, but mostly I'm a beachcomber. I'm a beach walker. And the beach is really this place of discovery of our ocean universe. And looking at the seaweeds, I hope after this talk, you might not walk around a batch of the seaweed detritus on the beach, but you might actually go and inspect it. It might be full of kelp flies and it might be a little bit smelly, but all of this is really great, a richness for the nearshore ecology. It's an incredible, all those insects are great richness to all the bird, especially migrating shorebirds. Think of all the protein that is being transferred, you know, into these insect bodies that then get to be eaten by shorebirds. And as you see here, there's this incredible array of shapes and colors and textures. So I want to get into some of the technical stuff about seaweed. It's hopefully not too technical, but the seaweeds, if you want to start kind of learning more about them, the place to start is what category they're in, what taxonomic group, what evolutionary lineage. And the seaweeds are broken into these three groups, the reds, the greens, and the browns. So the greens, and here it's another piece of ulva, the green seaweeds have just chlorophyll in their chloroplasts and driving photosynthesis. That's why they're so vibrantly green. And it's the green seaweeds that long ago migrated up into the near shore and then onto land and gave rise to our vascular plants on land. So it's the chlorophyll of the green algae that really is the chlorophyll that drives all the photosynthesis and primary production of our terrestrial plants. But our green is that chlorophyll is very good at processing red daylight. Daylight is predominantly in the red spectrum. But that's not necessarily the wavelength of light that's going to penetrate the ocean. And the red seaweeds, which evolved from the green many years, you know, billions of years ago, very early on, the red seaweeds have two accessory pigments, a red and a blue accessory pigment that collect different wavelengths of light from and then funnel that into the photosynthetic pathways. So those different wavelengths are what possibly are captured underwater. So that kind of resourcefulness is to be really amazed by. And then the brown seaweeds have a brown accessory pigment that combine with the chlorophyll to make these olive, golden, beautiful brown seaweeds. The brown seaweeds include the kelps. So the kelps are a subset of the browns, and any seaweed that has a bladder, a pneumatocyst, is in the kelp category. And the kelps are more differentiated. They're a little bit more complex in terms of how they're built. But they are still, they're all marine algae. This is a wonderful, this is a pressing and scan with its cyanotype of Maziella splendens. And Maziella is one of those just fantastic red seaweeds that you find in the intertidal zone or in the tide pools. And this is the seaweed that actually presents some iridescence in the tide pool. But when you dry it, the pigmentation from its chloroplast is what holds strong, and it's got a perfect combination of the red and the blue pigments that make it this perfect purple color. So I have a whole chapter on color in my book. So it goes into a lot more depth about the evolution of the seaweeds. Lynn Margulis, who was an amazing evolutionary scientist who is just a huge mentor to me. So I want to go into the story of the bull kelp, because the bull kelp, or Nereocystis lutkeiana, is the primary kelp of our Northern California coast. It is the canopy-forming kelp that grows from Santa Cruz or Monterey all the way up through Northern California, through the Pacific Northwest into Alaska and to the Aleutian Islands. And these kelp forests, I can't tell you how important they are. They are incredibly important as carbon sequesters, as sinkers of carbon. That's what photosynthesizing organisms do. They use carbon dioxide and transfer it and translate it into metabolic growth and off-gas oxygen. So they're great carbon sinks. They also are the primary producers. They're the bottom of the food chain, of the vast food chain and incredibly productive food chain of our nearshore oceans. And they are eco-engineers. They actually create habitat. So they are the places where so many other organisms thrive, not only because all that algal detritus sloughs off and floats around and is eaten by abalone and sea urchin, but there are all the snails and limpets that are herbivores and eat the kelp and the seaweeds. But it's also where predators hide, where prey hide from predators, where predators such as sea lions and sharks and whales and all that kind of thing go into the kelp forest to find prey. And there's a direct correlation between fish populations and kelp forests, the abundance of fish. They're also larval nurseries for many organisms. So it goes on and on and on. But the bull kelp, our dominant kelp, is an annual. It only lives for 1 or 2 years, and then it's wrenched up from the bottom and either thrown into the deep ocean or lands up on our beaches. So it starts in the springtime. So come this spring, February, March, April, May, look for the big low tides. Let's get out to the seashore and watch some of that algal growth. The bull kelp are subtidal, so beyond the low tide mark, and it will start as these tiny little organisms where the bladder is as big as my thumbnail. And these are some beautiful pressings from that UC collection at UC Berkeley. And in a matter of months, by June and July, this tiny little bull kelp will have grown to be 60 feet tall in some cases. And be this massive organism. And I hope you guys have been to the beach and have seen those big masses of bull kelp thrown on the beach and are stunned by the sheer biomass that they are. Well, that biomass is created by the power of the sun, the nutrients of the ocean, in just a matter of months. So here you have some pictures by Marco Pazza of the bull kelp in springtime as that bladder, that gas-filled bladder, is working to get the blades up to the surface, where of course the photosynthesizing will be much more efficient, closer to the sun. The bull kelp have this remarkable mechanism whereby once the bulb hits the surface, it stops the growth of this long stringy stipe, and the growth mechanism transfers to the blades. Now again, remember, this is only an organism that's going to live for 1 to 1.5 years. It's an annual. So it has to reproduce before the season is out. And so it will, by mid to late summer, start producing these spore patches called sori onto its blades. And that's what you're seeing here. And these spore patches get increasingly dense, and they migrate out to the end of the blade. And eventually all of the cells around the edge of it actually give way. And that spore patch then floats to the ocean bottom and disperses its spores. What then transpires is the alternate generation of this particular kelp, the Nereocystis lutkeiana, or bull kelp. It has an alternate generation that is microscopic and plays out under the ocean waves in the winter, in the dynamic and pounding ocean of Northern California, in the crevices of the rocks. So needless to say, it's not really been observed in the wild. It can be recreated in the lab. But what happens is those spores split and grow into either a male or a female And those male or female organisms are very microscopic and kind of filamentous, and they produce an egg and a sperm. And at some point, a sperm is released and it finds the egg. The egg actually produces a very a pheromone trail. So there has to be a sperm pretty close to follow that pheromone trail and fertilize the egg. And then those gametophytes then create the sporophyte, which is the giant bull kelp that we know. So this alternate generation, these very complex life cycles, is very typical of the seaweeds in general. The red seaweeds also have a very complex life cycle. But here's a picture of a bull kelp as it should be in late summer, where a lot of the spore patches have fallen away to the ocean bottom. This was from a snorkeling trip in 2017. And it was really wonderful. What I'm going to show you here is a time series of the bull kelp at Van Dam State Park in Mendocino. And this is where there's been a historic kelp bed. We have recordings of kelp here continuously over, since 100 years ago. But it is good to remember that bull kelp is very cyclical. So this is a picture from 2018 where it was at the very top of a cycle of abundance. And then bull kelp tends to go in these 3-year cycles of abundance, and then it falls away where there's not as much, and then it comes back again. So you can see what abundance there was in 2008. It's like, wow, this was the max of the max, an incredible amount of biomass there. In 2017, I was kayaking in that same historic kelp bed, and you can see how much more spaced out the kelp is here. And in 2019, when I went back past Van Dam State Park to give a talk up in Fort Bragg, I stopped and looked out over the bay there, and there was no kelp at all. And I think what's really struck me as I was sitting there was this, you can see it was this beautiful day, and the water was crystal clear and aqua colored, and the beach was white, and people were going off kayaking, and everything was beautiful. There was nothing to say anything was wrong. And it was only that I knew that there used to be kelp here. So I think this kind of how do we remember what we're forgetting is really very much part of my work that I'm doing now, this idea of historical ecology. So what has happened to the kelp beds of Northern California? And I hope at this point, this all played out between 2015, 2016, 2017, 2018. And then the journalists, and there have been quite a lot of stories. If you read Bay Nature magazine, there have been some really good stories on the decline in the kelp forest on the Northern California coast. But I'll just go over it very quickly. There's been a huge explosion of purple sea urchins. And what you have here is from that same snorkeling expedition, a beautiful bull kelp on the left, and then a bull kelp that has been trapped. It's actually attached to the bottom, and it's the blades have been pinned onto a vertical rock face by these urchins who are voraciously devouring its blades. And these kelp forests have actually kind of shifted from these healthy kelp forest abundant ecologies into these urchin barrens. Which have very little diversity to them. And so there were a number of factors. There was a huge explosion in these urchin populations because we had a couple of warming ocean events. And we also had the die-off of the sunflower sea star, who is a very effective urchin predator, or at least put some fear into the urchin to keep them from coming out. And the sunflower sea stars were only one of 2 top predators in the kelp forest system historically, and the sea otter being the other. And the sea otter, if you've been to the Monterey Bay Aquarium and around Monterey Bay where there are sea otter, you know how cute they are, but that they are voracious urchin eaters. And there have been no sea otter on the North Coast, or no sea otter north of the Golden Gate, and even north of Santa Cruz, really, for 150 years. They were killed off by the fur trade, and were really ecologically extinct by the 1880s, for sure. So we can't forget that they were a component, they were an evolutionary component to our North Coast kelp forest. So this was a piece that I made when really in 2018, when these, I was the grief, the tangible grief for the loss of these kelp forests was really very, very potent. So this is like the ghostly image combined with the fabulous crown of bull kelp by Alexander Postels. So now I'm just going to take you through a little bit more quickly. We'll just some fabulousness, kelp fabulousness. This is the Weeksia, named after the Mrs. J.M. Weeks. And if you buy my book, you'll find all sorts of clues left by the Mrs. J.M. Weeks. She was a wonderful collector in the 1890s for William Albert Sechells, who was the head of the botany department at UC Berkeley and is really the grandfather of California seaweed science. This is Postelsia, or sea palm. And again, I hope if you get out to the coast, maybe someplace like Salt Point State Park, at a low tide, if you look out, take your binoculars, if you look out at the rockiest wave crashed spots, you might see the sea palm. Sea palm loves that outer region where it will get crashed by the waves, it gets bent over, prostrate, and then it bounces back up. It's one of the seaweeds that really does look the most like a tree. So it's called the sea palm, or Postelsia palmiformis. And it got pride of place on the yearbook of the Minnesota Seaside Station in 1901. And then there was another one published in 1906. And if you, again, another fabulous woman pioneering woman scientist named Josephine Tilden established the Minnesota Seaside Station up on the west coast of Vancouver. And she brought all sorts of students and scientists to this remarkable spot to study the seaweeds. And Postelsia, its range goes right up to Vancouver, and it often establishes on mussel beds. It also is an annual, like the bull kelp, so it will rip up with its mussels. You can see it there. My specimen here actually has its mussel with it. And I think I write about Josephine Tilden in the chapter on coralline algae. And coralline algae are incredibly important to our nearshore reef systems. It often, the encrusting coralline, which you see on this bottle here, often creates this underlayer to the reef. So if you're out and you see this pink undertone to a rocky outcrop, that's really healthy. That's important. Larval organisms actually have there's some kind of mechanism that attracts it to the coralline algae to settle and then develop into its more mature self. So you have encrusting coralline algae, and you also have articulated coralline algae, which you have on the left here. Now, coralline algae does not grow very fast. It is actually calcifying in its cell walls as a strategy against herbivory, against being eaten. So algae are constantly, those leafy algae, it's very delicious to limpets and snails and all the little herbivorous organisms that exist out there. And the coralline algae have developed this strategy of saying, I'm going to make an armor against being eaten. I'm going to calcify. So Linnaeus, in fact, categorized the coralline algae in the animal kingdom at first, and then it got switched over into the algal kingdom. So here you can see quite a variety of different kinds of articulated coralline algae. It can be very, very delicate, or it can have these funny kind of wing nut shaped segments. Agarum is a wonderful holy kelp called colander kelp, and it really does grow with these holes. That's not from being eaten, that's how it grows. It's very prominent in Alaska, and it also evolved down the east side of the North American continent. So you will also find agarum in the Gulf of Maine, which is it's very, very few, there's very few crossovers, and this is one of them. And the story that I write about in the chapter of agarum is really about this cross-generational knowledge that comes from herbarium specimens. So these illustrations of agarum were from the very first flora written specifically about seaweed by a very precocious young scientist named Gmelin. And it was written in 1768. So Linnaeus wasn't that long since Linnaeus had actually established his naming system or categorizing system. And Gmelin used to describe, he needed specimens to describe from, so he used these specimens that had been collected by George Steller in the 1740s and had made the trip across the continent, the Russian continent, to the Academy of Sciences at St. Petersburg, and was sitting there in the herbarium, and Gmelin found them and used them to make these descriptions. And then a whole century later, actually in 1840, Postels made these fantastic lithographs about the agarum that they had collected in Alaska. Very incredible flouncy sculptural things. And he named this one Agarum gmelini. I have a whole chapter on the sea grasses, because these are very, very important as well as carbon sinks and eco-engineers and as primary producers. So the Zostra marina or marina or eelgrass is the eelgrass or the seagrass that inhabits the estuaries and bays, the shallower waters. And it is across the Northern Hemisphere and incredibly important. There's all sorts of restoration efforts going on, and a very successful one in the Chesapeake Bay. But the surfgrass or Phyllospadix actually evolved, and these are flowering plants. These are not algae. So those are rhizomes or roots here that you see. And these are flowering plants that actually recolonize the ocean waters from land. And the Phyllospadix actually recolonized the outer coastline, where it's pummeled by the pounding waves of the open ocean. That's where it likes it. It's a little bit thinner than the eelgrass. And there is a wonderful epiphyte called Smithora, an algal epiphyte called Smithora that will only grows on the surf grass, and its spores will only attach onto the thin blades of the Phyllospadix. So if you go out to Duxbury Reef in Marin, there's lots of surf grass out there. It's if it's coated with kind of a muck, it's actually this epiphyte that if you open out, it's very, very delicate. And if you can open up a little bit, you'll see it's that pinky red Smithora. These are some scans of the eelgrass. There's wonderful research on eelgrass restoration in San Francisco Bay that's going on over at the Romberg Tiburon Center, the Marine Labs of San Francisco State, that's actually titled the Estuarine and Ocean Science Center. And this is the lab of Kathy Boyer, who let me use these samples from her lab. And then I just have some pictures that is kind of my ongoing explorations at looking at the beauty of these organisms and how can I kind of indicate the abundance and the intricateness of the interconnections of the ecology that the seaweeds are part of. And these interconnections are really what we're constantly finding more and more about, and we don't even know the beginning of. So this is 5 foundational kelp, and this was actually a proposal for a mural in the redo of a lobby for the Hastings Law School, actually, and they have a good environmental law program. And I thought, wouldn't it be great for the law students to encounter 5 foundational kelp as opposed to any number of founding fathers? But that program went on hold with COVID, and I don't know whatever became of it. And then I've made a series of these kind of conglomerates that I title Ocean's Edge, and they're really meant to convey this incredible richness that's right here at the edge of our continent, at the edge of the ocean. And they're designed to be very big. They can be blown up as murals. I can produce them as architectural installations. I've done one commission for a stairway in a house. And so I'm hoping they can have all sorts of different applications, from websites to book to other architectural or public art installations. This was made all with my flattened dried pressing, so it has its own palette of color, and you can contrast that to this one, which is all made with wet pressings. So you can see that flouncy sculptural quality here, and it has a much different palette. It's a much earthier palette of all of these specimens that were put on my scanner really either right off of the beach, or I stuck them in my refrigerator for a day or so if I couldn't get to work on them right away, and then scanned them. And this is some of those abstractions that I really had fun playing with. Again, my experimentation with all of this color. So my project that I'm working on now is, I've been tentatively calling it Chasing Kelp. And it's very much looking at bull kelp in particular. And bull kelp is the second chapter of The Curious World of Seaweed. And so I want to expand from that chapter to look at what's happening with the bull kelp ecologies across its entire range. I've focused on Northern California, where I know a lot and I've written about, but also what's happening in Monterey and what's happening in Oregon and Puget Sound. And there's incredible work being done in Haida Gwaii and British Columbia. And what about Southeast Alaska, where bull kelp is actually extremely abundant? And then in Western Alaska. So that is where my work is heading now. This was a show at a great gallery in the Sunset District here called The Great Highway, where I kind of brought my collection. I wanted to bring the rack line from the beach, which is only a couple blocks away, into this wonderful storefront installation setting. And then one of the last things I did during this pandemic is I actually cast a piece of bull kelp, one of my kelp squiggle collections. And this is only a small casting. It's about 9 inches by 9 inches. And it bronze casting is very expensive, so that's about all I could muster. But one of my dreams is to be commissioned to produce a large scale kelp sculpture that is bronze, that can be climbed on and sat on and felt and have that tactile experience with, so kids can experience it and grownups can sit on it, and that tactile experience can kind of come out into a forum such as a park or a public space. So I'll end there, and I hope we have time for questions, but please really do be in touch with me. This is my email here, josieislin@lovingblind.com. I'll put this into the chat as well. I have a newsletter that I produce, my events page on my website. This is the books page here, josieislin.com, but the events page has lots of different presentations that I've done. I have a cyanotype workshop coming up right in Marin there at the Image Flow. You can find the registration for that on the events page of my website. I think it's /events. So please be in touch. And I hope we have time for questions.
Hi, Josie. I think I'm back. We do have we have quite a few questions. I just have to say thank you so much. And just, you've opened a door, I think, for me, and I'm sure others, to seaweed through your ability to see the beauty, the shape, the art, and the color, and all of this you've been showing us. It's just wonderful. It's very exciting. So there's questions about your scanner. How did you start to use your scanner in the first place?
Oh, wow. In the first place. So I got my MFA at San Francisco State. And when I got out of school, I was doing all sorts of different stuff. And I was applying for I was working with 2 other colleagues. We were applying for a lot of grants. And we thought, it would be useful to get a scanner. We were using all sorts of different material. I had a young baby at the time in a very chaotic, tiny apartment. And so we kept the scanner over at her house, her apartment, but I was, needless to say, doing lots of laundry, and I had this used dryer, and I was making this collection, had this circular dryer lint in it, and I'd clean off the dryer lint, and I was making this stack of circles of lint, and something possessed me to take that stack of lint over to my friend Kathy's house, where we had our scanner, and start scanning the lint. And I realized that the scanner just gave me this incredible quality. It picked up this nuance of detail, and I loved how it stripped away the context, and really the color was so true. So that was it. I started scanning everything around my house. It let me work through kind of the life with young children when you're trying to keep your art career going. And it was a very direct way of working. I didn't have to be in a dark room. And I really liked how it also gave me this workflow with Photoshop and InDesign for designing these books that I eventually were making. So that's kind of how it began.
There's a question about ocean pollution and how has that affected the abundance and variety of seaweed?
Well, ocean pollution, is not good for seaweed. It's not good for anybody. And it definitely, so the seaweeds are kind of a direct, because they take in the nutrients directly from the ocean around them, they are as healthy as their surrounding ocean. So there was a lot of, if you go back to the records of the seaweed collections of the 60s, a lot of it was done in the research around pollution mitigation. That was happening as people were realizing, oh, we shouldn't be pumping our sewage out into the ocean. So yeah, that question also has the reverse of how healthy are the seaweeds, and all the seaweeds are edible, but you don't want to eat seaweeds from unhealthy ocean, because it will be as healthy as the ocean around it. So most seaweeds are edible. There's no poisonous seaweeds like you have in the fungal world. You could take a bite out of, if you're, pretty much anything you find, especially if you're in a place where there's really great turnover of ocean. But there's certain seaweeds that people do. As I mentioned, the nori is a really classic edible seaweed that's eaten throughout the world. So it's kind of a cosmopolitan seaweed. So in Japan, it's nori, and here it's nori, and in England, it's known as laver. Forage, I'm not a big forager of seaweed. I think we really need to kind of really be conscious of where we are collecting, and we all need to know if we're at a marine reserve. But the people if you want to cook with seaweed, which I encourage you all to do, because it does make everything taste better. There's a wonderful, I would say, I think it's Daybreak Seaweeds. Is run by two women who've done tons of research. They wanted to start a seaweed farm, but California has made it very hard to do seaweed farming with all of their permitting process. And so they source their seaweed from farms that they've actually visited and are very confident in up in Alaska. And they're just wonderful. So I always have seaweed mixed in with my salt that I put into just about everything.
Fantastic. There was one person, maybe you could put this in the chat too, who wanted to know the full name of the 1840 Postals Ruprecht Folio at USC. But I think that's probably our last question. I think there's lots of comments about how fascinating, interesting, beautiful, and fabulous your work is. We so appreciate you coming to the Outdoor Art Club.
Well, definitely. I just put Illustraciones Elgarum was the name of that publication. If you're interested in finding it, email me. I'm, Josie Islin at lovingblind.com. I do put out a newsletter. I will be, if you're interested in any of the kelp recovery work, there's a lot going on right now. I can point you, if you're a diver, there's lots of really good people working on kelp recovery. So just be in touch.
Fantastic. Thank you so much. We really appreciate.
Absolutely.
The Outdoor Art Club's next public speaker event, Wildlife in Marin, Friend and Foe, will be held on Thursday, January 27th at 1 p.m. You can register for this event and all future webinars at the outdoorartclub.org under public events. Thank you for joining us. We hope to see you again soon.